Thanks, Marissa. Good afternoon or good morning, depending on where you are in the country. I titled this Alternatives to Animal Testing. However, it covers the FDA Modernization Act, known as 2.0, as well as the EU Directive 2010-63 EU. And for that, we will get into a little bit of the EU Directive since it goes further. So what is the Modernization Act? Well, in the way Congress passes bills, let us look at the Senate combined language, which I've put on the screen, because the Senate version actually summarizes in the best details the parts that I wanted to focus on today. So what you have is the old Section 505, the Food, Drug, and Cosmetic Act, which anyone who's done an NDA knows the 505B1, B2 pathways, the NDAs, BLAs go under a different law altogether. So what actually was changed? Well, within the law, in the paragraph known as 1A, you strike preclinical tests, including tests on animals, and you insert non-clinical tests. And in paragraph 2B, you strike animal and insert non-clinical tests. And then the Senate goes on, and this passed the House and subsequently was signed by the president. They defined the non-clinical test. And for biosimilars, they also modified a particular section of the PHS Act. So what ultimately happened, having been said once, was preclinical tests on animals became non-clinical and animal became non-clinical. Now, this is not a prohibition. You may have heard, oh, it's a prohibition. It is not. This is not. And this does not go as far as the EU law. The changes to the law are literally a few words, but those few words matter significantly. So what is a non-clinical test? According to Congress, for the purposes of this section, a non-clinical test means a test conducted in vitro, in silico, or in chemico, or a non-human in vivo test that occurs before or during the clinical trial phase of the investigation of the safety and effectiveness of a drug, and may include animal tests, or non-animal or human-based biology test methods. And we'll go into some of those in a minute. So it specifically says may include a non-human in vivo test. So again, there is no prohibition. It's still included. It basically expanded the definition. So as we looked at, there is no prohibition. So what is a non-animal or human biology-based test method? Well, it could be a cell-based assay. We see those frequently in R&D labs. It could be a microphysiological system, or it could be a bioprinted or computer model. So let's go on into in vitro, in silico, or in chemico. We all see in vitro on a near daily basis. In silico, computer simulation. In chemico, that refers to the reactivity of test substances towards synthetic model peptides, which generally contain either cysteine or lysine. The relative depletion of the model peptide is used to support the discrimination between sensitizers and non-sensitizers. That one may not be as most may not be as familiar with in chemico, but that's what it is. So what is a microphysiological system? We all remember physiology, but what this does is it uses microscale cell culture platform for in vitro modeling of functional features of a specific tissue or organ of human or animal origin. Now, the MPS design may aim to provide and support cultured cells with physical, biochemical, electrical, mechanical, structural, morphological conditions, and recapitulate a set of specific properties that define a healthy or diseased organ or tissue function. So this covers pretty much anything you could think of in terms of mechanical, structural. You could see devices show up in this. You could see tissues being built, morphology. And you could see or foresee diseased organs, diseased tissue. And so pretty much any condition could be replicated in theory. In reality, it's more complicated, but in theory, you could replicate. And these platforms may comprise monocultures, co-cultures of multiple cell types, maintenance of explants derived from tissues or organs, and inclusion of organoid cell formations. So having said a lot of words and used a lot of words in the law, let's take a look at one that has been developed at Vanderbilt with NIH support. We all remember molecules that either would or would not cross the blood-brain barrier. If you wanted the molecule to cross the blood-brain barrier, invariably it wouldn't. And if you didn't want it to cross, invariably it did. We all remember antibiotics. Certain ones cross, certain ones don't cross. It's why treating infections within the brain are so difficult, because a lot of your antibiotics don't cross. Fluoroquinolones come to mind as one class of antibiotics which do cross. And so what we have here that was built by Vanderbilt, it's a concept, but I'm sure by now it's come to reality, is a planar blood-brain barrier neurovascular unit chip. Basically, this replicates the blood-brain barrier, reducing the need for rodents, which were traditionally used. So let's fast forward to what is an organ on a chip. We've all heard of rodent on a chip or organ on a chip. So what is a real organ on a chip? Well, it is a microphysiological system, albeit a subset class, and it consists of miniaturized physiological environment engineered to yield and or analyze functional tissue units capable of modeling specific or targeted organ level responses. So now let's get to the nice pictures. I borrowed this picture to show experimental controllability and reproducibility. There is 3D cell culture. There's 2D cell culture. And in terms of experimental controllability, 2D cell culture appears to be better than 3D. However, in terms of relevance and complexity, of course, our dear laboratory mouse is highly complex. Yet, there are times that we have all seen where reproducibility of a mouse study was very, very difficult. and sometimes they could not be controlled for. We all have had, at one time or another, one or two mice or rats who defied the odds. They were supposed to get sick at 45 days, they didn't. They were supposed to develop symptoms at 90 days. They were kept in the lab, and 6 months later, they had no symptoms. And at that point, some of us will admit we used to name them. The mouse that wouldn't die, the mouse that wouldn't get sick. And then eventually, 6, 9, 12 months later, somebody would suggest looking to see what made the mouse almost immortal. And so we would, begrudgingly sacrifice the mouse and then start doing an analysis to find out what happened. That said, what do we have that mixes 2D cell culture for reproducibility and the complexity of a mouse? We have microfluidic organ on a chip. So in terms of increasing complexity, multi-organ microphysiological systems as tools to interrogate inter-organ crosstalk and complex diseases. Basically, let's put all these MPSs together and let's generate an artificial mouse or an artificial rat. And while we're at it, let's complicate it by trying to replicate a diseased rat or a diseased mouse in an attempt to replicate a diseased human. It sounds like science fiction, but as I've said many times before, science fiction and time and money, it becomes reality. And frequently it becomes reality very quickly. So as you can see here, we have the single organ MPS. We can couple two together and have a multi-organ. So then we can go from two, we can combine two, we can get four. Now, for biological material, what are the possibilities that we could use? What could we use? Well, we could use cell lines. We could even go up to tissues, circulating cells, organoids. And we could go to the most complex right now, which are 3D printed tissues. So how do you do the integration? Well, you've got immersed, you could use scaffolds, you could have them circulating, or you could have an air-liquid phase. For circulation, you could have static, passive or rocking, single pass-through. Single pass-through has its benefit when you want to study first-pass metabolism. Continuous, which is more the way the human body operates. Or custom. We build anything we can dream up. That said, what are the physiological cues? We can put pressure, replicate it. We can replicate architecture. We can use oxygenated blood. We can use deoxygenated blood, venous return. We can induce a charge, much as would be achieved with a pacemaker on the heart, but we can put charge. And while we're at it, we can complicate things even more by adding various hormones and cytokines. The more we go towards 3D printed tissues and barrier models and continuous or custom circulation and hormones and cytokines being present with oxygen-rich blood or venous return blood, we start to mimic the mouse or the human. So what can you do? What else can you do above and beyond that? Well, let's see. We could take 6. One for pancreas, one for liver, one for brain, one for lymph nodes, one for the intestines, and one for the kidneys. So if you had this setup running, you could theoretically, with intestine, kidney, brain, liver, pancreas, study a small molecule. And you could examine how well it crosses the gut wall, what happens with the first pass metabolism, and what are its effects, and the effects of the brain and the pancreas and the liver upon it, especially metabolism, when you have liver and kidney. Now, for that, let's go with crosstalk. And let's add into this cytokines, chemokines, hormones, metabolites, proteins. Let's add some immunity. Immune cell, adaptive immune, innate immune cells. And we have spectroscopy, multiplex assays, sequencing, flow cytometry, microscopy, all for analysis of the results. So you can take one small aspect and you can see this growing pretty soon into an artificial rodent or an artificial human. Where is this useful? Well, physiology, target and drug discovery, cell therapies, human-machine integration, PK/PD and ADME, safety and tox, unknown threat. We have a new threat out there, a new chemical agent that could be used in warfare. We have, any number of new pesticides, chemical pesticides that could be used. Personalized medicine, that's become quite popular. And vaccine design. These are just a few of the applications where these would have benefit. So let's complicate matters even more. Let's create a diseased donor model. Let's create the disease state. Sure, there's the control group. There's the naive mouse that's never seen drugs of any type. But let's create a model with 50% renal failure or 50%, liver function. Now let's create one that has both 50% liver function and 50% renal function. Let's create a model of acetaminophen overdose. That's doable. Let's create a neurodegenerative disease, or let's add 2 or 3 drugs and let's determine if we have antagonism or synergism. When this is pulled off, and this is becoming more of reality than science fiction, but when this is pulled off, you will be able to see pretty much anything you can dream up. If you dream it up, in theory, you'll be able to do it, and in reality, you probably will. So we can go further and to look at combinations and complexities with hepatocytes, stellate cells, neurons, astrocytes, microglia. If you just take 4 from the liver and 3 from the brain, you wind up with a 7-variable study. As you can see, all the interaction between just a little bit more or a little bit less of any one of the 7. And so this, coupled with artificial intelligence, will make preclinical testing of molecules very interesting in terms of the results that you could generate. In fact, this may generate more information than a mouse would. You couple it with machine learning and systems biology, looking at evolving networks, prediction, translation. You could run as much as transomics. And then you could come in with metabolic perturbations, hormonal challenges. And I just found the typo in the image, which managed to get published. So I only realized that at this exact minute. Immune perturbations and mechanistic validation, even up to as including CRISPR and siRNA. So let's look at an overview of this. What's the 30,000-foot view? The elimination of animal studies supporting drug approvals has been in the works for decades. This did not just happen overnight. Now, there is a big difference in, quote, not being required, i.e., under special circumstances where animal models are not predictive, versus guidance from the agency indicating animal studies need not be used to demonstrate safety. There's a big difference in those two. So where are the gains? What's going to be gained with this? Some might fear that animal studies aren't going to show up anymore. That's not the case. Your biggest gains are in two places. Carcinogenicity studies. The value of two species in lifespan studies is in question, and the FDA is using a weight of evidence approach to reduce some testing. And with personalized medicine on the increase, with humanized monoclonal antibodies and cell and gene therapies that are specific to human targets, the animal models are becoming less relevant and have been replaced in some cases by humanized animal models or tailored in vitro studies. This area will continue to evolve, and it may be difficult to get away from animal studies altogether, because what I showed you on basically the artificial mouse or the artificial human with a disease has yet to be totally validated. And so there's still much work to be done to get these to be commonplace. I'm not saying none have been validated. There may be some that have, but to get to the point where no single mouse is needed, we're a long ways off. We agree it's a laudable goal, and it would be the best outcome for various reasons. But we can't jump in feet first and just say we're not going to have any more mouse or rodent studies, or even non-human primates. So with that, let's look at what the FDA is doing, because at the end of the day, the FDA is going to be the judge of the MPSs and artificial mice and rats and non-human primates. So what is the FDA doing? Well, they have an alternative methods working group, internal. They discuss FDA-wide new in vitro, in vivo, and in silico methods for research, training, and communication. They interact with US federal partners and other global stakeholders, because at this point it becomes worldwide, to facilitate the discussion, as well as develop draft performance criteria for such assays. Because if the assay doesn't perform, and doesn't perform reliably, then it's just an experiment. And one that can't be replicated on top of that. They are working to establish a dialogue and develop the partnerships with the stakeholders to explore regulatory science applications, as well as identify the performance criteria of these MPSs talking to their own experts and stakeholders, and public and private partnerships. So the FDA knows they're moving there, but they have to do it in a systematic fashion. They do take input, and they do review what is submitted to them. Now, that's what's going on in the US. Let's look at what's gone on in the EU, because some of you may want to develop a drug that you ask for FDA clinical trial clearance, an IND, and you may want to go to EMA as well. So EMA goes even further. And their directive, which was now 13 years ago, further implements the 3 R's: replace, reduce, and refine. And we've all heard these, but they enshrine theirs in law to say replacing the use of animals with non-animal methods where possible, reducing the number of animals to a minimum with a caveat of scientifically valid results, and refining practices to minimize the stress and improve the welfare of study animals. If you have the slides, the hyperlink guideline on the principles of the regulatory acceptance of the 3 R's, and they have a nice reflection paper on how to reduce the animal testing. And they are hyperlinked if you get my slides. So what is the first R? That's replacement. So what would replace? Well, animal and human cell lines that have been established, cells and tissues, mathematical and computer models. What we just had in an earlier slide, the blood-brain barrier NVU chip, organ on a chip, multi-organ on a chip, and computer models. These are methods to replace. The second R is reduction. How can we reduce the number used while still obtaining relevant data? An underpowered animal study was a total waste. Yes, you reduced the number used, but your study was underpowered. So what can we do? We can improve upon experimental design and statistical analysis, combine studies. Instead of 1 arm and a control group, maybe it's 4 arms and 1 control group for all 4 treatment groups. International harmonization of the testing requirements, such as ICH, to avoid duplicate testing. This requires sometimes negotiating with both the FDA and the EMA. It's doable. It sounds difficult. It's just doable. It can be done, and you can get both sides to generally agree, especially when you say we're trying to harmonize. And imaging and other technology to enable longitudinal studies in the same animals. Now, what is refinement? Well, refinement, and we've all had rodents that could not be given anesthetics for one reason or another. Usually these are in CNS active molecules. But we do all work, and Europe enforces it even harder, to minimize pain, suffering, or distress or lasting harm experienced by animals. So with the EMA, the investigator has to make the case for why animals are needed. While it is not yet an outright ban on their use, basically requires non-animal methods of testing unless there is no alternative. The US doesn't go to that extent, yet we're running behind the EMA on this matter. There are some benefits, and I can see some large benefits as to why we would rather be behind the EMA on this. Let them go first, because while artificial multi-organ MPS, shown here, has its benefit and in theory works perfectly. Most have not yet been validated. And so there's a time that both will be needed. Yes, we're going to use some rodents to do it, but yes, we're going to work to validate the MPS. And so with that, I hope I have given you a good update of what recently passed Congress and was signed by the president on the use of animals in research and how we're working on creating artificial ones while still using animals and knowing that they are necessary, at least in the near future, if not for the next number of years. So thank you, and I look forward to any questions and or discussion.